S'il vous plaît, si vous pouvez donc éteindre vos micros. La main ديرهم بالواحد. Merci. On attend juste 1-2 minutes le temps que tout le monde arrive parce qu'ils sont en train de venir. Voilà. À vous la parole, professeur Bettayeb.
بارك الله فيك. السلام عليكم. رحب بكم جميعا في هذا الأمسية. Good evening, everyone. It gives me a great pleasure to be with you today in this series of webinars on advanced science and technology that the Algerian scholars and competencies abroad are organizing. Today we have the privilege to have with us Dr. Malika Aid-Boudries. She's assistant professor in medicine at the Center for Virology and Vaccine Research of the Harvard School of Medicine. Few words about the biography and experience of Dr. Malika. She holds an engineering degree in computer science from the Mouloud Mamri University of Tizi Ouzou in Algeria. She moved to Montreal, Canada in 2004, where she obtained a master's degree in immunology and molecular biology. You are hearing me, right?
Yes.
Yes. And molecular biology, followed by a PhD in computational biology and bioinformatics at the Institute of Research in Immunology and Cancer in Montreal, Canada. After her doctorate, she moved to the US for her postdoctoral training at VGTI, Florida, at Case Reserve Western University, and the Harvard School of Medicine, where she is an assistant professor in medicine. Her laboratory research focuses on using state-of-the-art omics technologies to understand the host virus interaction and explore diseases, pathogenesis, and immune responses to vaccines at the genomics and epigenetics levels to understand how the pre-vaccination host environment will influence vaccine responses and define the molecular correlates and mechanisms of action of vaccine. It gives me the privilege to give the floor to Dr. Malika to enlighten us on this very important, promising area of research. And the topic will be single-cell multi-omics technologies and data analysis methods. Dr. Malika, you have 45 minutes so that we can leave 15 minutes for questions and comments, please.
Great, thank you so much. Thanks, Dr. for the kind introduction. So good afternoon, everyone. I will skip the introduction slides, of course, and let's start just with a quick summary of my disclosures. I am part of these committees, and I'm also the CEO and the founder of Omics, which is a consulting company based in the US. So today, my talk will be more focused on how we can harness the power of omics technologies and omics data to study the host pathogen interactions and how the host immune response respond to vaccine or to any treatment or drugs. Actually, I will present my talk into 3 separate parts. So in part 1, I will focus more on introduction to omics data and omics applications. What do we mean by omics? Related to medical research, but also beyond medical research, because omics technologies and data are also used in somewhere else, in other research fields. The second part of my talk will focus on specific technologies called the single cell and the bulk RNA-seq profiling. And for that, I will first show the major differences between the 2 platforms and show a case study of how we applied single cell profiling in our lab. And the third part will focus more on omics applications to study the host pathogen interaction and the host immune responses to vaccines, showing some of our published work and other unpublished work from collaborators and also from our lab. So my slides are in English, of course, but please feel free to ask your questions in French or Arabic as you wish. And after each part, I will take a few seconds just to make sure that we are all set and you don't have any questions before I move on to the second one. So let's start with part 1 and what is omics data. So the definition, let's say the overall definition of an omics refers to everything that is related to large-scale data sets. And when we say or refer to large-scale data set, it takes high-throughput techniques and technologies to generate these high-throughput data sets. So the omics field include, but not limited to, genomics, which is the study of the genome, DNA, transcriptomics, which is the study of the gene expression from the genome. The gene can be, any gene at any certain time can be off or on. So transcriptomics look at this differential expression of genes. Proteomics, which is the study of the proteome, which is the result of genes. Metabolomics, which is screening or exploring the different metabolites associated with the host immune response, the response to a vaccine, to a treatment, to an infection, etc. Epigenomics, which is the study of the chromatin organization that is highly important and relevant actually to understand all the metabolomics, the proteomics, and the transcriptomics, and the genomics, because the way the chromatin is structured affects a lot how the genes are expressed or and immunomics, which is the study of the immune system in general. Microbiome, we know that microbiome is very important because certain microbes are very important and relevant for the, for any living organisms, and other microbes are harmful. So it's important to understand the microbiome within any living entities, including individuals, of course. Glycomics, which are specific sugar or proteins on the surface of proteins that are also highly involved in the way the host immune response, the host respond to a vaccine, to a treatment, to an infection, etc. And finally, the last but not the least, and these are really new technologies that that boomed this last few years, tissue imaging and what we call spatial transcriptomics and proteomics. And spatial here refers to the fact that rather than collecting your cells or your tissue samples from, from an individual or an animal models and process the samples, then do the omics analysis or assays, spatial transcriptomics and proteomics actually they keep the tissue or the cell environment rather than taking the cell out of its environment. We keep it there within the tissue and we study the tissue environment, including the cells within. Okay, so next, when we refer to omics in general, we have this a priori definition that omics is just about medical research or vaccines or treatments or drugs, but omics is also applied beyond medicine to many other important and interesting fields. So in agriculture, for instance, for food safety, food quality, how to design more resistant products, personalized nutrition, environment studies to look at the ecosystem and how what we do as human beings affect the environment. Forensic and criminal investigation, as you all know, we can use DNA in these specific cases. And omics approaches also are being widely used in what we call biofuel production or ce qu'on appelle les énergies renouvelables. Okay, so now let's focus now on, because this is what I do. I work more. My work is more in close proximity with infectious disease, vaccine development, the immune response to vaccination, and how we can dissect the interaction between a host. The host could be a human being, an animal model, and the viruses or the pathogens. So what do we need to conduct an omics project? Of course, omics projects or omics-based projects are cost a lot of money. So before you embark in any omics projects, you need to think about it and design, ensure that you have a well-designed study before you start with. So in this schematic, actually, I'm showing three major parts. The first one is, of course, if you want to do an omics project, you want to select the source of your data. Are you going to collect samples from a human being, from group of patient, or a healthy patient, or vaccinated patient, or a cancer patient, for instance, or use an animal model like mice or monkeys or any other species? Or you want to just use cell lines collected from one of these species. So this is the first step, the source of my samples. Once it's well decided, you want to design your experiment in terms of what am I going to collect? Am I going to collect tissues? Am I going to collect cells? Am I going to collect serum, plasma, cells, and tissues, etc.? Then decide whether you want to conduct a longitudinal study on different time points or just specific time points. So once you have all this decided and well designed, the second part is, given the data I have collected, what are the assays that are needed to process these samples? For that, the omics technologies are there to help you. So if you want to do bulk-seq, if you want to look at the gene expression at the bulk level, you can do bulk RNA-seq. If you want to look at the gene expression at the single cell level, you can do single cell profiling. If you want to look at metabolites, proteomics, cytokines, you can use serum or plasma, etc. So you need to understand the type of tissues or samples or cells you have in order to select the most appropriate or the correct technology for that. So, and the third part, and that is, it's an important part, not necessarily the most important, because the two previous ones are also very important. But the last part, what we call the integrative or the predictive models or the data analysis, the data integration. And this is kind of what we do as systems biologists or system vaccinologists, or if we want to call it bioinformaticians or computational biologists. For that, you need first a good understanding of the data you have generated. Because if you don't understand the data, you want to be able to to select the right or the appropriate tools to analyze it. So it doesn't take only someone who knows how to code, or someone who knows a data scientist who knows how to analyze data. It takes someone who understands the data and focus more on the biological insight, the questions you are trying to answer before selecting the right and the most appropriate tools to analyze, QC, analyze the data, integrate the data, and of course, evaluate and assess the statistical significance of your experiments, of your group comparison, of the efficacy of your vaccine or treatment. So these are overall the major component of an omics project. Select your samples, design the assay and select the techniques and tools, and then how am I going to integrate and analyze this huge amount of data sets? As you all know, or maybe some of you know, omics technologies are generating millions, if not billions of reads, of sequences. And it takes computing resources, strong and advanced statistics, and analysis tools for that. Once you have that and you successfully define these three major aspects of your experiments, now this is kind of a summary pipeline of how we analyze omics data to generate the data that your bioinformatician or your computational biologist can take, analyze, and test your hypothesis, because every single project has to start with hypothesis, of course. So first, sample collection, as we said, and it could be any of these type of samples. Then you need to process your samples using one of these, not only one, these are just some examples, of course, there are more than that. So one of these technologies, then once you have your assays set up and sequenced and data generated, then you take it to the analysis level where you can use free tools. There are many free tools out there for any bioinformatician or computational biologist to use. They are free. You need, whether they are in a format of R packages for people who are very familiar with R, or Python packages also, but there are also some web interface tools that you can use to analyze your data. Of course, one limitation with omics data set is the amount of data that is generated. So sometimes these tools are very limited and you will have to deal with your own resources to analyze it, to integrate it, or even to store the data on your servers or computers. The second way you can analyze your data set is in-house or ad hoc tools. For every single project, it takes a set of different tools or specific tools. Even though most of these tools are out there, you will have to take these tools from a more broad way of using them to make them more specific, to adapt them to your own specific data set. So don't think that if an R package is there, you can just go and take this R package and start analyzing your data. You will have to pay attention to what kind of data I have. Is this tool appropriate to analyze it, or should I do some tweaking? And this is also the job of your bioinformaticians or computational biologists if whoever is conducting these experiments is not very familiar with doing bioinformatics or data analysis. Okay, so this is the snapshots of some omics tools or pipelines. And this is really very restrictive, small snapshots of all the tools that are out there for omics data analysis. So you collect your samples from individuals, NHP, mice, or any other species. The first step is to generate the data using one of these technologies in these different squares. Then, depending on the type of data you have, these are some of the tools you could use to analyze it, to integrate the data, to QC the data, to make sure that the data, you can generate the expectation, what you are expecting from your initial hypothesis or experiments. So once you have analyzed, QC'd, integrated all this data set, and if your experiments actually is involving more than one platform, more than one species, you will have at some point to work in terms of, to think and work in terms of mechanistic models, because It's one thing to generate one specific omics data sets, but at the end of the day, what you want to do is how all these omics can be integrated together to answer my initial biological questions. So the main objective of an omics strategy is not the tools, is the biological question you have asked at the beginning, and if these tools can answer it or not. Well, assuming that everyone know what he's doing or she's doing, using the right tools, the right techniques, etc., and the data are generated, then we can start analyzing the data. So before digging into the analysis and giving you some specific examples, one aspect or one challenge in omics
Malika, your micro.
You are muted.
Weird. Okay, how about now? Okay, sorry for that. كنت تسمعوا فيها من قبل؟ Hello?
Yes,
Yes,
sure.
just for
Bien sûr, bien sûr. Merci.
Okay, parfait. Donc, the challenge is data integration. So why should we pay attention to data integration before starting digging, answering our biological question, hypotheses, and making sense of the data? Because omics technologies are very expensive and they are time consuming. So you want to make sure that your initial hypothesis, your initial experiments are well designed before you start like making them happen. So some of the challenges include, but not limited to, again, computational methods for power analysis and quality control. Once your data are generated, you need to make sure your data are good enough for downstream analysis. That's why you need always do some QC control on your data. And there are well-defined tools and standards for data and quality control. Also, power analysis. In the biomedical field, power analysis is very important because every single experiment starts with a hypothesis about the vaccine, about the drug, about a treatment. You want to make sure that the conclusion you will be making are correct, are valid, and can be applied on a large scale, right? So you can't just do an experiment on one person or 2 or 3 or 10 and conclude that, oh, my treatment is working. And that's what we call power analysis. And luckily, there are many tools that can help you do this power analysis and assess to what extent my conclusions, my findings can be generalized or not. For instance, I can do the following. I have a clinical trial where I collected samples from 20 individuals that received the vaccine, okay? And I want to assess the efficacy of the vaccine. 20 is not a lot, but in the context of clinical trials, it could be a good number. So what am I going to do? I'm going to say in my paper or in my grant or in my publication that using an N of 20, I am reaching or achieving a power analysis of 60%. You need to do that because otherwise people may think that, oh, this is a good thing, the treatment or the vaccine is working and we are all happy, but it's not. So power analysis is very important and you should be paying attention to that. The second aspect is account for differences across species. When you are dealing with omics data, sometimes you collect samples from one species, but if you are collecting samples from different species, you will have to account for differences, look to similarities and to differences. Both are very important, but you need to do some level of normalization so you can analyze and put your data together. Another aspect or challenge is account for technical and biological batch effect. When I say batch effect here, there are two main batch effects. There is the technical batch effect, which is due to the platform, to the way the samples were processed, and there is the biological batch effect, which is related to the differences between the samples. If you are collecting samples from cell line, PBMCs, and from tissues, you will have to correct for that, because otherwise you can't put them together, compare them head to head, because this is not correct. You will generate results, you will generate some conclusion, but they will not be true ones. Next, require imputation methods. And computer scientists on this call are very familiar with what we call imputation. Why? Because wherever you have large-scale data, we are dealing with missing data. And sometimes, when possible, you can just say, you know what, I'm going to ignore all the missing data. But sometimes you cannot, because this missing data can be very important, right? And can affect the way you analyze, the way you make your conclusions. So for that, you will have to account for missing data using, again, well-established standards and tools. Median mean, most frequent value, using the KNN, which is one of the algorithms we use in computer science for clustering, and look at the neighbors. If I'm missing a data at point X, I'm going to look at the neighbor of this X and take its value as the value of X, okay? So just so you know, there are tools and ways and methods to account for missing data. Normalization techniques before integration. Of course, when you measure for a protein in serum, and you measure a protein in plasma, and you measure a protein in tissue, the level of detection of this protein won't be the same, because these three type of samples are different. So in one of them, you will find maybe 10, a value of 10, in another one, in the second one, 100, and in the third one, 1. So the protein is there. It's measured by different techniques in three different tissues or samples. So you will have to normalize the data, bring all these measurements to the same level so you can compare them, right? So this is also a very important step in analyzing and dealing with omics data. Of course, reproducibility and data sharing, this is very important. You want to make sure that you can reproduce, or anyone else can reproduce your data or your findings. Develop the proper tools to cope with the rapid development of omics technologies. Omics technologies are evolving so fast. It's just crazy the way every single day there is something new, right? So tools has to evolve also and to adapt. And finally, resources for data storage and analysis. Given that omics data are huge, they require storage, of course, big storage, and it's money. When we talk about data storage, we talk also about money, and computing power, which is also related to money, because in some cases, for instance, single cell or tissue imaging, you will need very powerful computers or servers to analyze and integrate your data sets. Okay, this is about my part one, which is kind of, as I said, an overview of the omics. Part two, I will dig more into real cases of how I use this omics technology. This is just a small snapshot of one application of single cell profiling from one of our projects. So immune cell activation following vaccination using single cell profiling. So I'm not going to walk you through the study design, just so you know what we did. We collected samples from people who received the vaccine. It doesn't matter what vaccine. And we wanted to know, after vaccination, what are the immune cells that are activated by this vaccine? How these immune cells are evolving during the course of vaccination from day 1 to day 2 to day 3, etc. Okay, and before I show you the single cell data, let me just show this slide, which I took from the Cambridge Institute website. It's a very nice picture that depicts the differences between what we call bulk RNA sequencing and single cell RNA sequencing. So by both bulk RNA sequencing and single cell, we are trying to understand at any moment the gene expression. In bulk RNA-seq, we are looking at gene expression as a whole in the full sample. But when we deal with single cell, someone is using the cursor on the screen and making all these lines in colors. I don't know if you see. Okay, now it's gone. Thank you. Okay, so single cell, for instance, is looking into the single cell resolution. Look at it as very nicely depicted in this figure. I want a smoothie, right? You can get your smoothie and you don't really care what is in there. It could be banana, it could be any fruits, but I don't want the smoothie. I don't want to mix. I want to eat my fruits one by one. I want to eat my strawberries, my blueberries, my bananas, etc. So this is a very nice picture actually to explain the differences between bulk and single-cell RNA-seq. And as you can see, the resolution in the single-cell RNA-seq is very deep, is a high resolution, because you are looking at every single time point or in any single tissue or cell at one cell at the time. You can get the cell, sequence the genome of the cells, and say exactly what are the genes that are up or down in this specific cell, unique cell. In contrast, bulk-seq doesn't really matter about the specific or the unique cells. It takes all the cells together as a mix and tell you as an answer, as a response for your analysis, okay, you have these genes that are upregulated or increased in your sample, and this other set of genes that are downregulated, right? So it doesn't tell you in which cell these genes are expressed or which cell are expressing these genes, okay? Okay, so let's start with the some major known, well-characterized differences between bulk and single-cell sequencing. The cost. Single-cell sequencing and single-cell technologies actually are still very expensive. I'm giving you here some numbers from local numbers we deal with for every single project. To run a single-cell bulk-seq project, you need to pay around 200 to 400 sample for one sample. And for single cell, it's between 2,000 and 5,000, depending if you are running this in your lab, if you have a 10x, let's say, instrument, or if you are dealing with a core where you send your samples and they do the sequencing for you. Of course, single cell, because we are looking at a high resolution, it's labor intensive, it takes more time. It gives you, however, more details, more insight into the biology you are trying to understand. And it gives you also details of the cellular components of your samples. And it's more complex, requires more complex and sophisticated tools. If you remember my previous slide about all these challenges about normalization, batch effect, etc., if it's complex for bulk-seq, it's even 100 times more complex for single cell. So you will have to be aware of all of this and have the appropriate tools, statistics to analyze and make sense of your single cell data. So, and this is the case study I wanted to share with you today. So these are 3 samples. We ran single cell profiling on these 3 individuals, sample 1, sample 2, and sample 3. In sample 1, we sequenced around 8,000 cells and around the same number of cells in sample 2 and sample 3. We did our QC, etc. We normalized the data and everything was fine. Now I will be showing you some few figures from that. So identify the phenotype of the immune cell population. This is the major questions we asked for this specific project. We did single cell. I want to know what are the specific immune cells that are present in my samples that are activated by the vaccine. Important note, when we deal with single cell, we kind of not sensitive to power analysis anymore because we are sequencing cells. For instance, for this project, I have 3 samples, but I sequenced 9,000 cells. So it's powerful enough to make my conclusion and to analyze my data. And this is a very important point, by the way. So this is the first plot you get when you analyze your process, your QC, and you analyze your single cell data set. So the first step you do after you are done with the pre-processing, you need to estimate the number of clusters. I call it cluster for now, not cell type, because we don't know yet what are the cell types. So let's call them clusters. So from sample 1, sample 2, sample 3, because these are all individuals that got the same vaccine and we collected them at the same time point. They look very similar, right? The same cluster, you can see the same cluster at the same space, the same spot on the plot. So, and to estimate the number of clusters, there are again, tools for that, like the ones I'm giving here, just for your own curiosity, but there are many others that can help you estimate the number of clusters from the single cell assay. Now, I have my clusters. It's nice. I'm happy with that. Everything looks nice. But what are these clusters? Can I put a name on these clusters? To do that, you will have to know, you will have to have some basic knowledge in immunology and biology, because you need to know the markers that are expressed by every single cell type, right? And by doing so, you will be using these markers to annotate your clusters. For instance, here, I'm annotating this small cluster that is at the bottom of my plot across the 3 samples. I am using these markers. We don't see the names, but I can guarantee you these are all B cell markers. So I just took my B cell markers and overlaid them on my plots to see what are the cells that are expressing these markers. And it looks like this small cluster that is darker here on all the plots actually is the one expressing these markers, meaning that this is my B cell cluster. This specific cluster, I can call it now B cell, okay? And this is what you do with all the remaining clusters by using well-characterized and known cell markers. Okay, this is a more granular results from this single cell project. So in the previous slide, I showed you the 3 samples on different plots. And because they all came from the same clinical trials, from the same time points, I can put them together. And nicely, when you overlap the 3 samples, you get exactly the same plot, meaning that I was able to reproduce the same clustering, the same cell types or clusters using these 3 samples or 3 assays. Then, using the same approach, we annotated the different clusters. You have a cluster of B cell, of T cell, innate immune cells, including monocytes, NK, etc. So always remember to annotate your clusters. You will need to know the major or the top markers for every single cell type. Okay, after you define your cell type, you can look at the genes within this cell type, right? So, for instance, these are the major genes expressed in my B cell cluster. These are the genes expressed in my T cell cluster, etc. Once you have that, you can start having more fun and analyzing your data and making sense of it by doing all the fancy bioinformatics downstream analysis. Another application for single cell, so you can look at the overall composition of your samples, look at all the cells, but you can also be more specific and target a specific immune population. For instance, in this case, I'm showing a specific single cell project that define how B cells evolve or differentiate between 2 time points after vaccination. For instance, we collected samples at day 64 and samples at day 239. And you can see that when we look at different B cell population, there is this shift in the composition and the frequencies of the cells across time, which makes a lot of sense because B cells, there are the naive, the memory, the intermediate B cell, the plasma B cell, which are the ones that producing antibodies, etc. It's interesting to see that the shift from the day 64 to 239, you see more B cell producing antibodies later on, which makes a lot of sense. And this is the kind of application you can use, you can look into by using technologies such as single cell profiling. Okay, this ends my second part, and in the remaining, hopefully 10 minutes, I will walk you through some snapshots or templates project using multi-omics profiling, not necessarily single cell or bulk-seq. I will just show you the application, not the techniques. So the first project is a published paper, actually. It's about Zika infection. And if you remember, a couple of years ago, Zika was a major issue because of all these, neurological issues caused in newborns and children. So we collected samples from monkeys, we infected them with Zika, and we tried to understand. We performed bulk RNA-seq in this case, specific case, and proteomics profiling. So we used immune cells for bulk RNA-seq and serum for proteomics. And the major questions for this project was, can we identify some potential mechanisms of how Zika is infecting these cells and how Zika is affecting the immune system in general, but how Zika is creating all this neurological disorder or dysfunction in newborns and children. And this is what we did actually in this paper, and we were able to show that mechanistic target of rhabdomycin for the ones who are very familiar with this, this is a very important pathway, is involved in inducing or maintaining the pro-inflammatory state after Zika infection. So even after the virus is gone, because the virus has a cycle, it peaks around day 2 and day 4, and by day 14, there is no virus anymore in circulation. But we were able to show that the virus, even though it's cleared from the circulation, it persisted in in CNS and lymphoid tissues, and this is what was causing this persistent inflammation and all these defects and issues we have seen in a specific population, including children. The second project also published the study. Here we were more interested into the pathogenesis of COVID-19, or what we call the SARS-CoV-2. And again, we performed bulk RNA-seq transcriptomics, proteomics, and metabolomics. And we were able to show that during the course of SARS-CoV-2 infection in monkeys, this was done in rhesus macaques, by the way, we're able to show that there is this cytokine storm, which is well known and reported by many others, but we were also able to show that because of this pro-inflammatory state that is induced, highly induced after infection, there is the activation, there was a huge or a very significant activation of what we call the complement system and the platelet activation and aggregation. Platelets are important in wound healing and other immune functions, but in the case of SARS-CoV-2, there was this issue of thrombosis. And we found that complement activation and platelet aggregation are one of the major driver of this thrombosis observed in some patient with severe disease. Actually, patient died from the disease. The third project is about vaccination, and we have shown that actually, this is a specific project where we looked at the At26-CoV-2S vaccine, which is the Janssen vaccine, and we had clinical trials actually in individuals, of course, to look at vaccine immunogenicity and reactogenicity. But we also conducted studies in hamsters and macaques to look at the immune response of the host to SARS-CoV-2 infection, and how the vaccine if the vaccine is protecting or not. So in this very summary schematic, we show that in different groups of hamsters or macaque, when the hamsters or the macaque were vaccinated before infecting them, they were protected. How? We didn't observe this cytokine storm or a huge induction of pro-inflammatory or persistent of pro-inflammatory, pathological pro-inflammatory pathways. However, in what we call sham hamsters or sham monkeys, sham are the animals that didn't receive the vaccine, we saw this huge induction of pathological pathways associated with severe COVID-19 disease. So from this study, we concluded that giving the vaccine prior to infection protected these animals from developing severe disease, which is very important when we translate this data into clinic and into human being, of course. The other project I wanted to share quickly before I end my presentation is I'm personally interested in biomarkers discovery for disease, also for vaccine protection. And one of the projects I'm working on and keep working on actually is the HIV rebound. So just a quick summary about HIV infection. The issue with HIV infection is that the virus integrates in the host genome, which makes it difficult to be cleared, because once the virus becomes part of our genome and the cells that integrated this virus become silent, the immune system has no way to find them and kill them. And this is the big issue actually with HIV. That's why we don't have a proper treatment and we don't have a proper vaccine, because the virus is hiding. We will have to reactivate the virus so we can kill it. And the HIV-infected individuals actually take medications, and these medications maintain the level of the virus very low, so they can live normally. But these medications have a side effect, and patients actually end up by dying from cancer if they don't die from HIV. So there is a big research access now about what we call treatment interruption studies. There is an infected person. We give this person the treatment. But after a certain time, 6 months, a year, 2 years, we stop the treatment and we look at, is this person protected or is the HIV coming back or not? That's what we call treatment interruption studies. In this specific project, we are using, of course, rhesus macaque. We are not using individuals. So we infected them with the equivalent of HIV in macaques, which is the SIV. Then we put them on treatment for 6 months. Then we stopped the treatment and we looked at what we call correlates of rebound. What are the genes, the proteins, just before stopping the treatment that can be used as biomarkers to predict which animal will rebound or which animal will want to rebound? And by rebound, I mean which animal will have the virus coming back and in which animals we don't. This is very important because at some point, if we want to translate this ART interruption studies in humans, we need to know if we have to make the decision to stop the treatment, giving the treatment to XYZ, we need to know if this XYZ will have the virus coming back or not. Because if the virus is coming back, we are not going to do that. We rather keep giving the treatment to this person. But if we can decide and know that in this specific individual, there is this biomarker, if this biomarker is expressed at this specific level, I can stop this treatment, the treatment in this individual, and the virus is not coming back. Actually, this is a huge deal now in the HIV research field. And that's kind of the work we are trying to accomplish in this paper. Just a quick note on visual transcriptomics or tissue transcriptomics. This is also a new era, new research field in bioinformatics or in omics research, where rather than looking at immune cells or tissues, immune cells or serum or plasma, we take pieces of tissues and we do the assay on tissue at the single cell level by keeping the immune environment of the cell. So it's important to keep in mind that keeping the immune environment of the gene or protein is very important, because when you dissociate the gene from its immune environment, things change and it's not the same anymore. So yeah, with that, I think this ends the third part of my presentation. And this is just a few notes how we can, because I think this series of seminars are meant to create connections, networkings with Algerian institutions and Algerian researchers that are in Algeria, of course, with whoever is abroad. And I wanted just to touch base on some of the, the ideas we could work on as a group to reinforce these collaborations and maybe create a bridge between the labs in Algeria and our labs overseas. With that, I thank you very much, and I'm happy to take your questions.
Thank you, Dr. Malika, for this very enlightening and wealth of informative presentation that I am sure people already express in the chat the happiness and the usefulness of your presentation. There are more than 30 questions and comments, so I will choose only a few, that are quite general so that we get the maximum benefit. And for the rest, as you already mentioned, the purpose of these seminars is really networking and collaboration. And therefore, I'm sure you don't mind and you would be happy to receive any email from someone and you will be able to answer, right?
Of course, I will be more than happy to connect and answer any questions.
So I guess your email would be available, or you want to put it on chat, or?
I'm going to put it in the chat right away.
Excellent, excellent. So let me, maybe I'll start in case I have many questions, but in case there is somebody who would like to ask orally the question, I think we can entertain a few. Faiz, can we entertain some questions if they want to speak?
Yes. So please, if there are,
Yeah, is there anybody, let's see, participant, anybody raising his hand?
Just raise your hand. Yeah.
Raise your hand, please, in the system. If you have, if you don't, if you are not raising your hand, I will go ahead and read some questions. So let me ask Dr. Malika the questions that we're repeating many times. One of the questions is, what is the results of the application of artificial intelligence to omics data from point of view of what do we use it for classification? Is it mainly just machine learning, or where is the difficulty all around AI for omics?
Perfect. This is a actually, I'm highly interested myself. I'm doing some AI research in my field. So AI needs one thing among others, big data. Big data is the best fit for AI. And we do have big data with the omics. We can generate billions of data and we can train our models. We can create, not only train and feed the model, we can give the resources, we can give the appropriate data to the model to learn and create the decisions by itself. And in the field of biomarkers discovery, actually, you can use AI a lot because AI is about discovery, is about prediction, is about how the system will respond to a new type of data or new information. We can use it also for prediction. For instance, there is a huge now domain of application from bioinformatician, computer scientists, or AI, experts on how to develop these devices to detect, I want an AI that tells me if I am susceptible to have a heart attack. Just by connecting the device to my hand or to my arm, sorry. So I think application of AI in omics is just beginning. And with the help of biologists, immunologists, physicians, because it takes good understanding of the data and how we use them, computer scientists, AI experts, we could achieve a lot. AI will play a major role in the kind of research we are doing now.
Right. Yeah, a second question that is repeated by many, how is omics used in renewable energies, in particular renewable energy production, for example?
Well, this is not necessarily my field of expertise, and I think I will defer it to whoever is working on this field in the audience to answer. I know omics apply to medical field. I know that omics can be also used somewhere else, but I don't think I am the best person to answer the use of omics dans les énergies renouvelables, unfortunately.
Okay. Another question is, what are the computational tools and algorithms and software used in omics?
Okay, this is a very good question and a very broad question also. As for now, there are at least 1,000, if not more than 1,000 of tools, of software, of R and Python packages you can use for omics analysis. But remember, omics is not one pool. Omics is about the technique you are using to generate the data. If you are dealing with bulk RNA-seq, you will have to look to tools that can analyze bulk RNA-seq data. If you are doing proteomics, there are different tools to analyze proteomics. Metabolomics, there are also different tools to analyze metabolomics. That's why there is a diversity in these tools. They are not perfect. And every single day, there is a new tool at least that is on bioRxiv or on PubMed or somewhere else. Why? Because the more data we generate, and the way the omics technologies are evolving, there is this continual need to develop new tools. Use what is out there, because most of these tools are free, especially for R and Python packages, but keep in mind that there is room for more development in terms of omics tool and omics analysis and omics integration. So I can, please, whoever is interested, you can email me. I can guide you based on what kind of data you have, because there are, as I said, thousands of tools. So let's look first to what type of data you want to generate, so I can give you exactly the appropriate tools to use.
Excellent. In fact, this was the next question. You answered it already. Somebody asked, can we need your free tools, most common tools you are using, and therefore they can contact you and get it. Somebody...
Yeah, so in addition to the tools, sorry to interrupt you, Dr. Tayyab,
sure.
The tools, most of the tools are free and you can use them for free from R or Python, but there is also something that is more important. Most of the omics data, the ones at least I have presented today, and also from other labs, are also free. Not the list of genes or the protein, the raw data, the way they came out from the sequencers, whether it's 10x or Illumina or other technique, the raw data and its requirement when we publish a paper, the journals require from us to put this data public on specific databases. One of them is the NCBI databases. And I think NCBI is free for everyone. So if you want to get familiar with omics, go to NCBI database, download the data, go use these free tools, play with the data, look at it, see, at least get a sense of how it looks. What is this omics? I want to learn the type of data that is out there and how to analyze it. And you have this opportunity for free, basically. So yeah, you can do it.
Excellent, excellent.
Professor,
another question is, what is, yes, please.
Yes, there is someone that would like to ask directly the question.
Okay, yeah, we are ready to listen.
Yeah, so it's Aid-Boudries Malika, if you want to ask the question.
And she has the last name, same last name. Hi,
Rima.
Rima.
Hi. First, I want to thank you for the amazing presentation. We learned a lot, I'm sure. But my question was about the data, like the type of models for data analysis. Like, would you rather use a unsupervised or supervised model, or a reinforcement or semi-supervised? Like, what are your go-to when you're doing data analysis?
Very insightful question, Rima. Actually, it depends on your biological question or your initial hypothesis. What is your initial hypothesis? If you are comparing A to B, if you know your targets, I'm looking to this specific gene, to this specific pathway, then you can use supervised tools. If you are going into a fishing expedition, you have this huge omics data set, but you don't know what are you looking for. Now, then the unsupervised approaches will be much better, much suitable, because it will give you an overview of what is about the data, the component of the different components of your data set. Then you can choose and select. So these questions are highly important, but they are really data specific. So it really depends on, first, your hypothesis, your questions, and what are you trying to achieve by your initial hypothesis. But all this is in omics, okay?
Perfect. Thank you so much for your answer.
My pleasure.
Great. Another question is, what is omics data in healthcare?
Okay, so we can look at it both ways. Like some of the projects I presented today, these are all data from the healthcare, right? These are data from clinical trials. These are data from vaccines, from infection. But the other aspect of omics in healthcare, this is this boom also in using AI to develop these devices for disease detection or disease prediction, or even organize healthcare data. Actually, just a week ago, I was talking to someone who is at Harvard, actually at the Reagan fairs. This group of young students are developing a chip using AI to put together all the healthcare data associated to one patient and try to commercialize this idea in countries where the health system is not as developed as in the US or Canada and Europe, because it's just one chip where you have all the information there and you can share it with doctors, with the hospitals, etc. This is one use of omics actually in healthcare, in addition to the research aspect of healthcare, of course.
Great. Another question is, what is the difference between genomics and omics?
Okay. So genomics is just one small part of omics. Omics is the big circle, if you want to call it that way, and genomics is a small circle inside.
Another question is, what are omics biomarkers?
Okay, omics biomarkers are all the genes, the protein, the metabolites, the glycans, the proteins, I said proteins, that you can generate using omics technologies to predict an outcome. Let me give you a clear and a straightforward example. I want to know, are there any biomarkers that can predict Alzheimer's disease? I can collect serum, plasma, CSF samples from a group of all the individuals, do all my omics assays, including metabolomics, proteomics, glycomics, generate the data, analyze the data, integrate the data, then look at the expression of these markers and see how they can correlate or not with the evolution of the disease. If I can find these good correlations and associations using machine learning, of course, then I can call it a biomarker of disease. And the biomarkers actually are widely used in cancer. And people working on cancer, physicians on these calls, know that there are at least, maybe I'm wrong, but at least 100 of kits that are out there you can use to predict cancer. Not necessarily specific cancer, but there are kits, biomarkers, 2 genes, 3 genes, 4 genes. And you take the kit, you do the test, and you can say, oh, this specific individual has a predisposition to have this specific cancer with a probability of 8%, 90%, 90%, 100%, etc. So this is kind of one use of biomarkers discovery using omics.
Professor Batayeb.
Okay. Yes.
There is someone else that would like to ask directly the question.
Excellent.
So Nasima Amara, you can unmute yourself.
Assalamu alaikum.
Alaikum assalam.
First of all, I want to thank you, Professor, for this presentation. My question is about how can we use omics to study and understand antagonism phenomena in microbiology?
Well, do you want me to answer? I will answer in English. Okay. So the antagonism, you know, the word or the mechanisms is broad. So can you give me like a more specific question? Because studying antagonism, la notion d'antagonisme, elle n'est pas spécifique. Are you referring to a specific field or application, or is it like a more general, you know, question? Microbiome, you are referring also to microbiome, right?
Yes, yes.
So, عندك une question spécifique على l'antagonisme ولا حابة تعرفي غير على l'antagonisme en général?
Non, en fait, je veux savoir l'antagonisme chez les, par exemple, on prend l'exemple des bactéries bacillus vis-à-vis des champignons, par exemple, vis-à-vis des champignons. Est-ce que comment peut-on utiliser l'omics pour savoir et étudier
Parfait. Donc, je ne suis pas un spécialiste des bactéries, mais je répondre à la partie omics. Donc, si j'ai bien compris ta question, les bactéries ou les pathogènes, ils sont dans un corps. Donc, لازم الحاجة الأولى اللي ديريها collecting samples. Collecting sample depending whether it's a serum sample or a plasma sample or even a gut sample from the gut, because this is where the microbiome is kind of se fructifie. Donc, du coup, start by collecting your échantillons, your sample sets. Then you do if you are looking at a specific pathogen or microbe, then you can do a targeted analysis using maybe not omics, a more targeted assay. But if you want to look at the broad overview, you can do microbiomics, which is an assay that you can do, by the way, to look at the full microbiome in your sample. You can also do some sequencing analysis to look at the sequence of your microbes, because knowing the sequence is also interesting. And this is also part of the omics, by the way. You could do gene expression and see how these microbiomes are affecting the immune response, because there is a tight link between microbiomes and genomics and transcriptomics. So email me, why don't you shoot me an email, and we can
Thank you so much.
discuss it more in a more specific way.
Excellent.
Ok, je serai très reconnaissante. Merci.
Merci beaucoup.
Yeah, and
Thank you.
somebody has a question. It's in agriculture, really, but let's see if you happen to have seen this kind of application. He's saying, I'm looking for new products for saving fruits and vegetables in storage areas. Does omics can help in any way?
Actually, I think this is a very interesting question, by the way, because what's the issue with storage of food is les produits, ils chassent après un certain temps. Donc, du coup, si j'ai bien compris, la question, c'est comment utiliser l'omics pour étudier comment protéger les produits et assurer la durabilité de leur storage, si j'ai bien compris la question. Donc, en fait, oui, vous pouvez faire ça. Pourquoi? Parce que si vous séquencez le protéome, le transcriptome, le génome de ces produits-là, vous pouvez le comprendre un petit peu, peut-être en faisant des extrapolations avec d'autres études. Qu'est-ce qui fait que ce produit-là, il résiste longtemps, il résiste à la chaleur, il résiste à l'humidité, il résiste à certains environnements, et un autre produit, il ne résiste pas. Donc, by doing this kind of analysis and comparing them, you can kind of, I think, get to a point where you can say, oh, this specific parameter is important because it makes this specific product last longer. Je pense it's a very good way of using omics, by the way. I really think that this question is very interesting.
Excellent. Yes, please.
There is another question, maybe
Excellent.
one of the last one is from actually Dr. Shidna. So if he wants a question.
Professor Ahmed is a specialist in this area. Let's listen to him.
Okay. Assalamu alaikum. Thank you for nice presentation, Malika. So my question is, since that the cost is very high for single cell research and analysis, how to lower the cost so developing country can use it?
Perfect.
This technology. That's my first question. The second one, how can we use single cell analysis for cancer diagnostic, for example, and treatment?
Perfect. Okay. So for the thank you so much for this insightful questions. For the first part, I would say that there are actually techniques that to lower the cost of a single cell assay, what we call hashing. So usually what we do with single cell, you collect samples from different individuals or time points. You decide how many cells you want to sequence because the cost depends on the number of cells. Whether you sequence 5,000 cells or 10,000 cells, the actual cost, the one I gave you, around 2,000, 5,000, is about 8,000 to 10,000 cells. Let's say you want to reduce the cost lower. We can do hashing. Hashing means I want to take few cells from sample 1, few cells from sample 2, few cells from sample 3, put them together and get to my 8,000 cells. Rather than paying for 3 samples, I will be paying for 1 sample. So we call it hashing in the single cell field. So hashing is combining single cells from different samples that came from the same experiment, the same time point, etc. Of course, we need to make sure of that. So you will, if you have 10 samples, you will reduce the cost by 10 factor of 10. Other than that, I think it's more developing maybe more technologies, because for now, single cell is kind of in terms of single cell technology, there are at least 5, 6 maybe major technologies that do that. So maybe having more companies doing single cell and developing new tools for single cell will reduce the cost. And even Algeria, why not? We could, a company from Algeria could invest in this kind of development and have their own single cell sequencer. For the second part, single cell, one of the major issues among many others, of course, of cancer is the heterogeneity. Cancer is not the same for patient 1, patient 2, patient 3, patient 4. Why? Because there is this interaction between the cancer cell and the host immune response. So by doing single cell, I think single cell is providing a huge jump in cancer research in general for the last 5, maybe 10 years, because rather than looking at the overall transcriptome or genome of the tumor, we can look at the specific cells within this tumor, and we can do the same for the host. And depending on the tumor environment, actually the gene expression, the cell surface markers, the host environment that is surrounding the cancer cell can affect the way we target this cancer cell, we kill this cancer cell, but also can affect the way the treatments are working. So in a more broad answer, I would say that single cell will allow us to target the cancer, the specific cells, cancer cells, and so can the immune cell, so we can kill them by developing the appropriate, medications, drugs, or treatments.
Good. Let me ask the last written question, and then we'll see if there is any other oral questions. The last question, I happen to be interested in this because I do signal and image processing. The question by someone is stationary wavelet in image processing effective in single cell analysis?
What is the name? The first
stationary wavelet. Wavelet transform is a tool in signal and image processing to do analysis, so decomposition and everything.
I do think so, because what we are doing now, similar to what computer scientists are doing for image analysis and image synthesis, to dissect the image and detect patterns and detect the specific component on a specific image or figure, we do the same on what we call tissue imaging. So you dissect your tissues, you take a slide, what we call a slide of tissue, then you can kind of take the tissue slide as a picture, as an image, and you process it using the same tools used in computer science. And this is kind of the beauty of all this applications and integration between computer science and tissue imaging, I think. I would say yes, all the tools that are used in imaging, analysis, and synthesis in computer science can be translated to tissue imaging and omics imaging analysis.
Mm-hmm. Let's see if there are any people who would like to ask questions. Dr. Faiz, is there anybody who would like to ask question orally?
No, there isn't. And yeah, I think it's also the time to.
Yeah, I think it's the time. We really thank you, Dr. Malika. It's really great pleasure. Very enriching, very informative. And as you said, this is just the beginning of hopefully communication between you and all the participants. So we thank you so much for your outstanding presentation. And we thank all the participants, all the colleagues with us here, and Dr. Faiz, of course. You want to close, Dr. Faiz? For me, we thank everybody, and we look forward to see you coming Saturday, same time, for another webinar that is already listed, titled in the chat. Thank you all.
Thank you so much for having me. It was a great pleasure. Salama.
Great. It was our pleasure, really.
Thank you very much. So
thank you. Thank you all. Mahiddin, barakallahu feek. Shukran.
Assalamu alaikum.
Shukran. Shukran. Shukran.